Mitra Pratape from Atomic Energy Central School number 4, Mumbai. Today I am going to teach you the practical about the study of different water samples for pH, clarity and presence of any living organisms. As we know that water is an important abiotic component on the earth and it can change its quality because of addition of substances, organic, inorganic, biological or radiological or factors like heat which degrade the quality of water so that it either becomes health hazard or unfit for use is called water pollution. Most surface waters usually contain small quantities of suspended particles, organic substances, a number of living organisms like bacteria, protists, fungi, algae, viruses, etc. The concentration of these substances in water bodies increases, then the water becomes polluted and unfit for use. Some other substances pollute water even at low concentration, like poisonous or toxic chemicals and pathogens. The presence of organic waste in water promotes algal growth, resulting in the depletion of oxygen in water. This is called as eutrophication, and this causes the death of organic organisms, that is because of the lack of oxygen. And there are examples in which many ponds and lakes they have become dead. They have been declared dead because of the growth of the algal blooms in the water or the aquatic body, and that has resulted in the eutrophication. Now, water pollution due to organic waste is measured in terms of biological oxygen demand. It is the amount of dissolved oxygen needed by bacteria to decompose the organic waste present in water. Higher BOD or the biological oxygen demand of water indicate presence of microorganisms in large number due to the presence of high concentration of organic matter. Now this experiment we will be going to do in three different parts. First, we are going to check the pH of the water samples. The second, we are going to do the experiment in which we are going to check the presence of particulate matter or the suspended pollutants. And the third experiment is the presence of any living organisms which are present in the water bodies. So the requirements for this experiment is, the first experiment we are going to do is collect water from two different water bodies around you and study them for pH. The requirements for this particular experiment is water sample A which we have collected from tap. The second one, water sample B, that is collected from a pond. The pH paper, that is broad range, and the forceps. Now we are going to start the experiment.
Start.
Now for this experiment, first of all, we are going to take the broad range pH paper. This is the broad range pH paper. Here you can see the color and the pH which is written against it. Now pH paper is very sensitive, children, so we have to use a forceps for taking the particular slip of this pH paper. We will take the pH paper, holding it with the forceps and then dipping it in sample A for a few seconds and then removing it from the sample and placing it against the broad range pH paper, the scale. Now here we have to match the color of this pH paper which is attained by it, and we have to match it against the shade card. So slowly we can see that the pH of this matches with 7. So this water sample that is collected from tap water, it contains pH 7. So this is the correct pH of the sample A. Now this can also be done with narrow range broad pH paper, in which we can get how much pH, accurate pH is that of the sample A. So same way we are going to repeat for the water sample B. Again we are going to take the pH paper and we will be dipping it in the sample B, which has been collected from a pond. Dip it for some time. Lift the pH paper. Let it dry for some time. And then you can match it against the range with a shade card. And then you can see that the pH of this aquatic body is somewhere near 8. And again, we can match it with the narrow range pH paper to get the accurate number. So this way, children, we have tested the pH of the two water samples which are collected from two different sites. Now, how you are going to record the observations of this particular experiment. So, this is how we are going to record the observations. This is the observation table for recording the pH of that particular water sample. So, we make 3 columns. Column 1 where the serial number is given for the number of water samples like 1 and 2. The water samples A and B. The pH that we had done for water sample, it is 7, and the pH for water sample B, it is 8. So this way we record the observations. Now the precautions for this particular experiment. We have to take the pH paper and after dipping it in the water samples, we have to dry before we compare it with the given shade card. That is the first precaution. The second precaution is the color should be matched very carefully to determine the pH accurately. And if you want to take the water sample in some other glassware, that glassware should be dried. Suppose you are taking the water samples in the test tubes to check the pH, then the test tubes should be dried. If you are taking the water sample in the Petri plate, then the Petri plate should always be dried. So this is the first part of this experiment where we have tested the pH for the two water samples. Now we will be doing the next experiment of this particular practical.
Start.
The second experiment in this, collect water from two different water bodies around you and study them for clarity and presence of particulate matter. The requirements for this particular experiment is the water sample A collected from tap and water sample B collected from the pond, the torch and the cardboard box, which is a closed box, and we have made two holes, one on this side and exactly the other on the other side, and this is the open region of the box from where we can keep the beaker inside. So we will be starting the experiment now. First, we are going to perform the experiment with sample A. So we will keep the beaker from the open end in the box, and we are going to close the end so that no light enters in the box. This way we have closed the box, and on the other side we are going to put the torch light so that the torch light can pass through this, and from the other end we are going to observe the water sample which is kept inside and to see the particulate matter if it is present in this. We can see that the water is clear of this water sample A. Similarly, we are going to perform the experiment for sample B. Similarly, we are going to keep sample B inside the cardboard box and place it between the two holes and close the lid. We are going to place the torch on the other side and check for the suspended particulate matter, and we have to put our eyes on the other side of it. Here we can see some particles are suspended in it. In this suspended particulate matter is C. So this shows that the water is not clear and this water is not potable. Whereas the sample A, we had checked the clarity and it was clear. So this shows the water sample A is clear, whereas this water sample B is not clear. The observations can be written like this. The suspended particulate matter, as I have already told you which are the suspended particulate matter, like the fungal spores, the bacteria, etc., can be observed in the sample B because we have seen it in sample B, whereas the sample A is crystal clear. So we can also mention that in sample A, this observation of suspended particulate matter was not there, but in sample B, it is observed. The precautions: the hole which is made on the cardboard box, it should be small, it should not be large, and it should be at the same, you can say, the region on both the sides. The light source should be of sufficient intensity. So this is our second part of the experiment. Now we will be performing the third part of this experiment. So the objective for this experiment is to study different water samples for the presence of living organisms. The requirements for this is water sample A, water sample B, glass slides, cover slips, droppers, forceps, compound microscope. Blue and the spirit lamp is optional, children. If you want to fix the slide, we can use the methylene blue and the spirit lamp. But if you don't want to fix the slide, then we don't require the methylene blue and the spirit lamp. Now, how we are going to perform the experiment? Let us do that. First of all, we are going to prepare the slide for sample A. You have to be very careful, children. We have to use separate droppers for different water samples. So this is for sample A. We will dip it in the water sample and take only one drop of this water sample and place it over the slide. Remove the excess of water. Use a forceps for holding the cover slips. Gently place the cover slip over the drop of water. As we know that the tap water does not contain any microorganisms, but still let us observe it under the microscope. So place it over the stage and clip it and observe under the low power, that is 10x. Now here you will see as the water is clear, so you cannot see any particulate matter or the living organisms present in this. Similarly, we are going to prepare the slide for sample B. Now, taking the other forceps for sample B, we are going to dip it in the water of sample B and take a little amount of water in the force dropper and take only one drop of water. And we can spread it also, and we can if we don't want to spread and want to see the organisms live, children, we can directly put the cover slip over this drop of water and observe it. So let us observe it first, because at times we see beautiful paramecium moving in the water sample. There are other organisms also which you can see moving here and there, so you feel happy after seeing those small organisms in the slide. So this way we are keeping it under low power first and observing it. So here we can see the fibrous particle which is present here, and along with this, children, you can see the moving paramecium, rotifers, beautiful microorganisms which you will be able to see in this particular slide. So just now I have shown you one video in which you could see some moving organisms which was done at some particular day. Because the slide was very good, so I had kept it for this particular experiment. Now these are the different types of organisms that you can also observe in the pond water. Now the precautions for this experiment is that, children, first of all, you have to be very careful regarding the droppers that you are going to use. Different droppers should be used for different water samples. The mixing should not be done. Otherwise, if some microorganisms are present in pond water, they will get mixed up with the tap water or any other water sample that you have taken. And the second thing is that whenever you are taking any particular water by the dropper, it has to be shaken well, and then you have to put it on the slide. And the heating, if you're making a smear of this, then the heating should be done only once, not more than that. So I think you must have understood regarding the particular experiment how you are going to perform. And these pictures, the diagrams which are shown here, you can draw it under observation. So this will be your observation, children. For sample A, because it is taken from tap, there will be no microorganisms seen in that, as we have already seen. And in sample B, you can see few microorganisms if it is having large number of organisms present in it. So diagrams can be drawn for observations. So thus we end for this particular experiment. Thank you.